Welcome to the front row. My name is Jamie Williamson. I'm a professor here at Scripps Research in the Department of Integrative Structural and Computational Biology, but I'll be your host today, and it's my pleasure to serve as the moderator for Hugh Rosen's lecture. Now, I did want to point out something. If you didn't notice the large 100 that's on the stage, that's happening. So yeah, this is our Centennial, and so it was in 1924 that the Scripps Metabolic Clinic was founded by Ellen Browning Scripps, and we can trace our roots back to that. In fact, we share a lot of history with the Institute for Oceanography and the Scripps Hospital, but we've been separate for quite some time, so we no longer have those affiliations, but we do share common roots in the philanthropy of the Scripps family well over 100 years ago. So there's a lot of events. The Centennial is the opportunity to see where we are and how did we get here, and also to take a look at where we think we're headed. And I think today's lecture is very much in the spirit. So there'll be some other events that you might be interested in tuning in for or coming to. And so we will certainly keep everyone posted about all of that. Okay, so today we're going to talk about making a drug. So I looked up some numbers. So the pharmaceutical industry in the United States spends about $100 billion a year on drug development, R&D. And that's matched by a pace of introducing new medicines at a rate of about 30 to 40 per year. So that means for every drug that comes on the market, about $3 billion got spent. Now, that doesn't mean it costs $300 million for each of those, or $3 billion for each of those, but because many of them never make it. So there's this problem that lots of drugs never even make it to the market. So lots of people discover compounds that they think might be drugs, and very few actually succeed in seeing those come to market and be helping patients. And so today we're going to hear from Hugh, and between Hugh and his partner Ed Roberts, their lightning has struck twice. So they've invented and they discovered and pioneered the ozanimod drug, which is approved for multiple sclerosis and ulcerative colitis. Now, today, we're not going to hear about that story today, but this is the next story. So probably that compound came out of their labs maybe 8 to 10 years ago. And so then it goes off to development in the industry. So what have they been doing lately? Okay, that's what you're going to hear about today. Let me advance. Whoops, that's backwards, forwards. So the theme here is precision medicine. So what does that mean? Basically, drugs have to undergo clinical trials, and they're basically set up so that they treat as many people as possible. But there are really some narrow indications where you can use what we call precision medicine to develop drugs that target some particular indication. And this is the subject of today's lecture. It's been particularly difficult to develop drugs for brain disorders, and there's a variety of reasons for that, and there's a lot of people that suffer from various problems. The issue is that the brain is incredibly complicated, and we've heard in a couple of these front row lectures that there's hundreds of millions of different kinds of cells. There are all these receptors, which are the targets of drugs in the brain, and they're in different places in the brain, and they have overlapping functions in different places. So it can be very difficult to narrow in, like a laser beam, with precision medicine on brain disorders. So we're going to hear today about how, using some just clear basic thinking, you can advance a very particular strategy. And we're going to hear about targeting the Kappa opioid receptor. And so today, Hugh is the chairman of the Department of Molecular and Cellular Biology. I'll let him introduce himself. And So I want to just welcome everybody who's virtual. I think we've got about 1,000 people online, and we've got about 200 here in the audience. We encourage you to come. So Hugh's going to give his lecture. I'm going to shut up, but then I'm going to come back at the end, and then we'll have a little discussion. And if you raise your hand, there's going to be a lightsaber that will indicate where you are, and raise your hand, and then I can point to you. If you're online, you can type at any time during the lecture a question into the Q&A. And then I'll get those up front, and we'll take questions from both the virtual and the live audience. So at any rate, please welcome Hugh Rosen. All right.
Thank you very much, Jamie, for the introduction. One of the things that I do is I stand here as a representative, not of myself, but of my colleagues, Ed Roberts, and our combined lab groups, where the heavy lifting has gone on to help discover these mechanisms and translate them into the beginnings of meaningful therapeutics. So let me begin by acknowledging them, and then I shall acknowledge them in detail at the end. Translational therapeutics is a challenge that goes back to the beginnings of medicine. But in modern terms, I like to dwell just briefly on some of the key thinkers in medicine that have influenced really the way I think about translation. The first is William Osler, who was at Hopkins and then was Regius Professor of Medicine at the University of Oxford, who was a typical 19th century rationalist. He looked upon disease as originating, in a sense, from mechanical failure. Disease was an expression of the broken machine. If you could fix the broken machine, well and good. But there were many instances where you could do absolutely nothing. And so the key to his approach was good physical diagnosis and a knowledge of the life history of disease so that he could care for the patient in the best way possible, whether it was, in fact, the treatment of their disease or, in fact, the treatment of their soul in the process of the disease. He was succeeded in Oxford by Archibald Garrod. Archibald Garrod was the physician and geneticist who first discovered the inherited defects of metabolism, a term that he coined in a paper in 1908. And in an essay that he wrote in 1910, he changed the way we thought about disease by showing that disease could be the result of what he called a maladapted evolutionary product. Disease was a combination of genetics and environment, and the selection of maladapted products led ultimately to the expression of disease. And with remarkable foresight, in 1910, he referred to disease as having its origins in the chromosomes from which we sprang. And this was 34 years before Oswald Avery showed that DNA was in fact the means of genetic transmission. So remarkably good thinker about disease. Howard Florey was professor of pathology at the Duns School in Oxford. And I have a particular affinity for Florey for two reasons. One is that in his work with Ernst Chain to turn penicillin from the product of the penicillium mold to a safe and effective human therapeutic, he taught us about the notion of transformational intervention in a disease when we understood the basis well enough. The affinity comes from the fact that I trained as a graduate student in the lab in which Florey had grown the penicillium molds in bedpans. And it was always an inspiration to think that something that came from a bedpan could turn into a transforming event in infectious disease. And now, over the past 10 years, we've seen at Scripps the notion of science changing life, where Pete Schultz's framework and his efforts have allowed us to scale translational therapeutics in ways that we could never have dreamed were feasible in the academic setting in any time. And this really has been a transformation in the way that I and many others have looked at therapeutics and how we're also able to catalytically act in the discovery of therapeutics. And so I'm very grateful and fortunate to be standing before you today to talk about one such episode of discovery. The great physician and president of Memorial Sloan Kettering in New York, Lewis Thomas, in an essay in the New England Journal of Medicine in 1974, it's called The Medusa and the Snail, wrote that the mechanisms of disease are quite open to intelligent intervention and reversal whenever we learn more about how they operate. And the story that I want to tell you today is how we've stepped through the puzzle of intelligent intervention, how we've been able to find those finger posts that point us towards successful therapeutics by digging into problems and by not giving up when we run into roadblocks that seem to be insurmountable. That inductive logic can really get you there. This is my colleague, Ed Roberts, who has been really integral in both the discovery, invention, and development of ozanimod, and now, of course, navacaprant. So our goal has been to use the power of chemistry and genetics to define and to alter nodal points of control in human disease. Can we define these control points that can be shifted ever so subtly to create a useful impact on the patient and on the disease process? One of the reasons why this has worked at Scripps have been its inherent advantages. Firstly, we've had good research leadership. Secondly, we have a group of colleagues. We have minds that think deeply and are committed to understanding problems in the greatest of depths. And these are also open minds that collaborate and contribute so the whole becomes greater than the sum of the parts. We have the infrastructure, because that's always absolutely essential in practicing science and technology at the highest levels in the modern age. We have the knowledge that gives us depth in our understanding of pathways. So we may see things that other people will miss, or our colleagues will see things that we have missed, and together we make that difference. We have to have the powers of persistence, because nothing is easy, and it's a struggle, and it takes many years, often decades, and it takes much longer than you think, and it takes much more resource and effort than you would ever imagine. And none of this could actually happen without the administrative and technical backup we have within the institution. The scientist is the tip of the spear, but the hazardous waste needs to be taken out. The labs have to be air-conditioned. Everything needs to function in a way that permits safe and effective scientific conduct. Patents need to be filed. Patents need to be defended. The interface with the federal government needs to be managed very, very closely. And so without our administrative and technical support, we also would not get this done. So moving on to the subject of the day. And I wanted to start with really a few basic definitions so that we're all on the same page. And the first is, what is anxiety? So anxiety is a feeling of fear and dysphoria, feeling bad, that mimics the feelings of fight or flight and often occurs in a non-physically threatening setting. It has mental components, namely the fear or dysphoria. It has physical components that result from the overproduction of stress hormones in the periphery, such as adrenaline. Sympathetic nerves getting input from the brain via the spinal nerves innervate organs such as the adrenal glands. They produce catecholamines, adrenaline and noradrenaline, and they're overproduced. The heart pounds, the heart rate speeds up, blood pressure is elevated, there's hyperventilation, we blow off carbon dioxide, the plasma alkalinity rises, ionized calcium drops, and in the midst of a flamboyant panic attack, hands and toes claw up in cramps. It's really quite unpleasant. The triggers are many, and these behaviors, because they are learned and follow triggering and amplification over time, become autonomous. And then we deal with generalized anxiety disorder, an anxiety disorder that is frequent and non-reflective of the stimuli from the environment. Depression is the comorbidity. It goes often with generalized anxiety disorder. And the American Psychiatric Association defines it as a common, serious, but treatable illness that negatively affects how one feels, the way one thinks, and how one acts. Depression causes feelings of sadness and a loss of interest in activities once previously enjoyed, which is called anhedonia. And more on anhedonia when we discuss the clinical data on the Scripps compound. There are changes in appetite. There's trouble either sleeping too little or too much, loss of energy and fatigue, an increase in purposeless physical activity, pacing, hand-wringing, or slow movements or speech. People feel worthless or guilty. They have difficulty thinking, concentrating, or making decisions. We call this brain fog. And some people have thoughts of death and, in fact, of suicide, which is a very, very bad place to be. So if we think about where to start, and it's sort of a bit of a detective story, which is why I have Jeremy Brett as Sherlock Holmes to remind me that this is, in fact, the way we piece things together from fairly small clues. We start with what the unmet needs are in a condition, in this case in psychiatry. And I wanted to focus here on diseases with thought disorders. So mental stress-related and developmental disorders are both undertreated and have a high prevalence in the population. 18.4% of US adults will have a lifetime diagnosis of depression. 2.8% of US adults had a past year prevalence of bipolar disorder, which is depression coupled with a thought disorder. The prevalence of schizophrenia ranges from 1/4 to 0.64% of the population. The challenge here is that 50% of schizophrenic patients, for example, are non-adherent to therapy. So although 80% can be stabilized by first-line antipsychotics, around half of all patients don't stick with therapy. They're particularly at risk of non-adherence if they've newly started therapy, if they're either younger or older at the time of treatment onset, if there's a background of substance misuse, and if there's poor social and familial support. Disease-related drivers, if there's high levels of cognitive impairment, poor insight, and psychotic paranoia, patients are less inclined to be able to be preserved on therapy. And then the medication drivers include the side effects and the complex treatment regimens. So what then are the common side effects of the first-line therapies for both depression and, in fact, thought disorders? There are sedation, there are abnormal movements, gynecomastia, weight gain, cognitive impairment or brain fog, sexual dysfunction, and then the usual dry mouth and difficulty urinating that goes with agents that are cholinergic in their activities. So now let's switch to thinking about how we go from an unmet need to the basics of mechanism and then back to attempting to solve an unmet need. And so I want to mention here the yin-yang seesaw of endorphins and dynorphins. Most people are familiar with endorphins. There are some people, and I pity them, that get a high from exercise. That is a result of the production of endorphins that signal by binding to one of the opiate receptors, the mu receptor that binds morphine and related drugs. Endorphins stimulate the pleasure centers of the brain and deliver a sense of happiness and well-being. There are also those people who don't like exercise and for whom exercise, in fact, may be a dysphoric experience. And those people are wired more to the production of dynorphin. Dynorphin is an endogenous opioid ligand, much like the endorphins. It is produced from a protein precursor called prodynorphin by proteolysis and is elevated in its production by the stress response. It's elevated in the brain in anxiety and depression. It activates the Kappa opioid receptor and induces anxiety, agitation, bad feelings, impaired executive decision-making, and as I shall show you, also hallucinations. And when produced by chronic stress, it helps convert anxiety into depression. So this is what we call the ligand. Dynorphin is the ligand. And the ligand then acts on a protein, a receptor, to switch responses on or off. So let's just think briefly about cell surface receptors. These are proteins that are integrated within the membrane of the neurons within the various areas of the brain. They bind specific molecules. For instance, the mu receptor will bind morphine with very high affinity. They are the sensing switches of the cell. They recognize the ligand. In response to the binding of the ligand, either the peptide or morphine, for instance, they change shape or what we call conformation, and they switch on. They transduce a signal that amplifies the response and transmits that response broadly around the brain. The receptors themselves then have mechanisms of terminating the signaling or, in fact, switching the signal off. So we refer to ligands that activate receptors as agonists, the on switch, and ligands that block the receptors are antagonists, or the off switch. Now, in the matter of opioid receptors, they're part of a massive receptor family called G protein coupled receptors. In fact, there are 701 members of the class A G protein coupled receptors, like the opioid receptors shown up here. And there are about 68 or 268 others that bind more complex protein ligands. Now, on this little node of the receptor dendrogram, where they are laid out by DNA structure, there is a small family of 4 receptors. And 3 of those are the Kappa opioid receptor, that's going to be the focus of my talk, the mu opioid receptor, that mediates the action of morphine, heroin, and fentanyl, the delta opioid receptor and the opioid-like receptor, neither of which I understand adequately to talk about. So the first family, the morphine receptor, was found in the early 90s in terms of its structure being cloned. And out of that cloning, they defined by homology these other 3 receptors that we see. And we focused on the Kappa receptor for one particular reason. We thought that the Kappa receptor was a stress receptor. It was really good if you wanted to escape from a saber-toothed tiger, but is absolutely appalling as a part of modern life. And the reason for this is actually quite simple and complicated in a number of ways. So the Kappa receptor is shown here as the white K on a pink background, and this is a sagittal section through the human brain. And the focus, the areas that are highlighted, are in fact the pleasure centers of the brain. So areas like the nucleus accumbens, the ventral tegmental area over here, these are areas that produce a lot of dopamine, and dopamine is the neurotransmitter that stimulates the pleasure pathways. So these areas are dopaminergic and they stimulate pleasure. So if you would say, what is the source of the gratification associated with sex, death, and rock and roll? You would say the dopaminergic areas at the base of the brain are exactly what do that. The dorsal raphe nucleus over here, which is rich in the Kappa receptor, is an area of serotonergic signaling and is actually the site of mechanism of action of the common first-line antidepressants, the serotonin release inhibitors, as well as the noradrenergic and serotonergic release inhibitors. So the SSRIs and SNRIs act here on the dorsal raphe nucleus. What's fascinating is that the Kappa opioid receptor regulates a variety of neuronal circuits that impact on emotion, cognition, addiction, and stress and dysphoria. So we thought it was a really good candidate to play around with it. I'll show you later that the emotion and executive function reside in the prefrontal cortex and in the connections between the prefrontal cortex and, for instance, the amygdala, and how the Kappa receptor can influence our ability to make good or bad decisions. So it's a complicated system, but these are fundamental signaling mechanisms and nuclei that are at the center of our emotions, our gratifications, our motivations, our decisions, and how we therefore behave. So how does this work? So shown here is the Kappa receptor, shown here as a G protein coupled receptor in the plasma membrane of an inhibitory neuron. So you have dynorphin being produced and secreted. It acts on the Kappa receptor, shown here in the ventral tegmental area, and this is a dopaminergic neuron. And what Kappa does is it stops the secretion of dopamine. So Kappa activation by dynorphin induces the presynaptic inhibition of dopamine. So dopamine is no longer secreted and then no longer stimulates the pleasure pathways that are required for emotional and other satisfactions. Dynorphin, as we mentioned, is produced in response to stress. So Kappa activation by dynorphin inhibits dopamine release presynaptically, and this is bad because it down-modulates pleasure, satisfaction, motivation, and normal movement. So how do we know that this is disease relevant? Firstly, we know that dynorphin is upregulated in brain areas that are associated with mood and reward, and we know that that's not good. Secondly, we know that it induces dysphoria, anxiety, and depression, as well as hallucinations in both human and animal models. So therefore, blocking it should be good. And disruption of signaling through the use of genetic knockouts or receptor blockers have antidepressant and anxiolytic effects in animal models. So let's look at some of the pointers, the finger points, the finger posts that pointed us to this target. And the first is a warning. Some lines of scientific evidence arise from people doing stupid and extremely dangerous things. I'm going to show you a video from YouTube. Please do not try this at home, because the line from social experimentation to addiction or psychiatric illness can be exceptionally short in some individuals. There is a weed, a sage species called diviner's sage, Salvia divinorum, that grows in parts of Mexico and South America. And the natural product active is salvinorin A. This is an isoterpenoid that actually activates the Kappa opioid receptor highly selectively with mind-bending effects. So here is a human social volunteer participating in a social experiment where he has smoked the leaves of Salvia divinorum.
Where are you at, brother? Where are you at?
He begins to see things. He begins to hear things.
It's all good.
In the space of
Hey,
seconds,
you're okay.
he is overtly hallucinating
What do you see?
and has had his reality completely
Wi-Fi?
changed.
We got plenty of
They
Wi-Fi.
have a sense of overlapping realities,
I don't know, bro.
paranoia, and
Why-
hallucinations.
It's okay.
These are quite remarkably
all here.
rapid in onset. They impair judgment, disrupt sensory and
Watch out,
cognitive
Joe.
functions. And unfortunately, Salvia divinorum is
far as the
legal in
chair.
39 of 50 of these United States. So whilst the DEA does just talk about its drug facts, it's not been made illegal, despite the fact that it can cause very severe hallucinations. There is other evidence that we know that the Kappa receptor drives psychosis. Aniline was a selective Kappa receptor activator that was developed for pain relief, and tiny doses were tested in man. The doses starting at 1/33,000th of a Tylenol tablet. And at doses that were that tiny, subjects were reported anxiety, dysphoria, abnormal thinking. And the dosing had to be stopped because 100% of the subjects started hallucinating. So we know that highly selective agents drive hallucinations. So our hypothesis was that if we could selectively block the Kappa receptor, we should block anxiety, it should be non-sedating, it should disrupt the progression between chronic anxiety and depression, it should interrupt processes involved in psychotic behavior, and that we could test this by directly making a reversible blocker of the Kappa receptor. So 10 years ago, we didn't really overthink how we were going to do this. We simply did a small chemical screen on a diversity set of 16,000 compounds in the library. We had a compelling hit that inhibited the activity of the Kappa receptor. And even though this molecule was not especially attractive, we have a wizard in the house. And that is that all of our compounds have a prefix that begins with CYM, which is a reference to Ed Roberts' origin as a Welshman. The mythical magician Merlin has his origin in Wales, where he is known as Merddin Wilt. And the secret to therapeutics is it doesn't matter how smart anybody else is, but you really need the right magician doing the chemistry. So here was the starting material straight out of the Maybridge Diversity Library set. It was half micromolar, not particularly exciting, and most people wouldn't even take it that seriously. We have a process that we call rapid multi-parameter optimization. We don't do things the way most drug discovery organizations, whether they're biotechs or pharmaceuticals, do it. We do it in reverse. And that is, we think about this in two ways. We have molecules that need to interact with high energy with a target, but we're only interested in the ones that will work extremely well for the purpose that we're after. So we don't think about the end point, we think about the beginning point. We strip our hits down so that the energies converge to a minimalist core scaffold. And then we ask the question, what are the factors in those scaffolds that will impact on interactions with people? What is the nature of the core scaffold? Does it have good toxicological properties? Will it distribute in the right way into the right compartments of the body? We don't worry about the interactions with the target, namely potency and selectivity, until later. And the reason is that interactions with people are sort of a crapshoot, whereas the interaction with the target are always rational and are driven by an understanding of chemical and protein space. So take the rational parts out of it. Those we know we can do. Let's deal with the irrational parts, which is always interactions with people in any which way you might want to look at it. And what we try to do, therefore, is go from thinking about free energies to threading the needle through interactions with people and with the target, and out of that, selecting a compound that is good. But every potential drug has to survive near death. The process is really difficult, and you've got to be robust enough to weather the death zone, and these are the accidents of biology or funding that can kill you. We have to deal with funding risk. Without funding, you have withering on the vine. We have hypothesis risk because we truly don't know what we don't know. And that's the risk that has to be taken to believe and push something forward. And then we have technical risk, the properties that are rational and can be resolved by inductive logic, as I've mentioned. But you also need to get a compound that's working on a psychiatric disease into the appropriate parts of the brain. And both distribution to the brain and efflux out of the brain are properties that are a little more random to deal with. So you need to manage technical risk for the parts you can logically and for the parts you may need to brute force. So success requires finding the right inductive path to a solution that works. And I'm just going to give you one example. Mutagenicity is an unacceptable cancer risk. When a compound is mutagenic, either on bacteria in the Ames test or in the micronucleus test on eukaryotic or human cells, it disqualifies that compound from development because it may represent a cancer risk. Now, one of our early leads was in fact Ames positive on a single bacterial strain. When that happens, investors run the other way. Companies that have such agents immediately stop development. But Ed and I ask questions. And the strain that was involved with this Ames was only a single strain. And it was a strain where, in fact, we knew that the lesion was, in fact, a micro-insertion, where there was the addition of one extra base pair into the DNA sequence. So we asked, can we deduce an answer if the solution is rational, or is brute force the only hope? Given that we had very limited resources, it had better have been amenable to logic. And one of the things that we reasoned was that insertion mutants require binding to the minor groove of DNA. And we thought that the methylquinolinoamine metabolite predisposed to minor groove binding. So we asked whether we could change the chemistry in a subtle way to kick the molecule out of the minor groove and retain excellent receptor activities. So shown here is CYM 50202, which was Ames positive. It was 12.6 nanomolar on the Kappa receptor. 51317 was also Ames positive on the Kappa, in the Ames test, but had good other activities. We made two minor modifications, the fluorine in the 8 position and the methylation here, to kick the molecule out of the minor DNA binding groove. And lo and behold, we ended up with a molecule we now call navacaprant that was 0.8 nanomolar on the Kappa receptor and highly selective, and has been clean enough and good enough to persevere in clinical development. For a molecule to be a drug, it has to work every single time it comes off the shelf. You can't have activities that are not robust. Shown here is just the affinity plot, the Schild plot, where a competition curve is done in our automated assay, looking at the right-hand shift induced by the antagonist in the activation curve, and allows us to measure the affinity. And this has to be highly reproducible and precise and work every time. Otherwise, we have no interest in it. It also needs to work in a variety of orthogonal assay formats. So shown here is navacaprant. Its radioligand binding to the Kappa receptor, its selectivity, right? 300-fold selectivity versus the morphine receptor, and then binding against a variety of ligands. There are two dynorphins, dynorphin A, dynorphin B, that are the product of proteolysis of prodynorphin. So we need to be able to block the dynorphins. We need to be able to block all of the signals generated by the Kappa receptor, because we want a clean and competitive neutral antagonist that simply switches off the Kappa receptor without any further complications. It also needs to work in every animal experiment, and here I just show you that we have a 2.5-hour mouse experiment where we look for the secretion of prolactin in the peripheral blood that is a response to the suppression of dopamine in the hypothalamus by dynorphin, and that was allowed us to determine that compounds work. They work orally, and they cross the blood-brain barrier. And so in 3 to 4 days in every week, we were able to know all of the receptor activities and all of the in vivo activities, including brain penetration, with massively rapid turnaround time in order to be successful. Early compounds can be discovered and happen in academia, but developing them into drugs obviously takes hundreds of millions of dollars. Optimization, selectivity studies, preclinical toxicology. You then have to file an investigational new drug application with the FDA, and then 3 clinical phases. Phase 1, which is safety, phase 2, which is blinded efficacy, and then phase 3, which is safety and efficacy in multiple trials that allow a compound to reach the market. So where have we got with navacaprant? So at that time, we really only had one choice. We didn't have an internal option within Scripps, so we formed a company called Blackthorn Therapeutics, now acquired by Neumora Therapeutics, that allowed us to do much more additional work, that taught us more about the wiring diagram and progressed the compound through early clinical development. And just mentioning Christina Burrow at Arch, who persevered with this compound through numerous difficulties to help progress it to where it is today. We have a variety of other evidence that suggests that the Kappa program will have important interactions in additional elements of human disease. Experiments with our colleague Frank Poreka at the University of Arizona showed that our Kappa receptor antagonists actually work on the inhibition and on the on the prevention of migraine allodynia in rodents. So it does appear to be able to blunt some of the elements of allodynia, pain that is experienced in the central nervous system. But the most interesting study from Frank and his colleagues was a study in rodents looking at neuropathic pain. So they looked at chronic neuropathic pain where you damage the sciatic nerve of a mouse, and it then recruits a hypothalamic dynorphin kappa opioid receptor signaling, and the output of this chronic pain is in fact wakefulness and vigilance. And what we found shocked us. Navacaprant restored rapid eye movement sleep in chronic pain. Now, sleep disturbance, for those people that grapple with psychiatric illnesses, is a symptom of anxiety, of depression, and schizophrenia, and is a tremendously tough side effect of first-line therapies, such as the SSRIs. So the ability to both impact on anxiety and depression and impact on sleep disturbance may actually be quite a remarkable benefit from patients. So navacaprant went into phase 2, and I'm just showing you one piece of the phase 2 data that I pulled from Edgar, one of the Neumora corporate filings associated with their IPO. But navacaprant was dosed for 8 weeks, shown here in moderate to severe depression patients, and again randomized versus placebo. And the Hamilton D17 is a complex scoring mechanism of both psychological and physical symptoms of depression. And what you can see is that within 2 weeks, the curves begin to differentiate, and navacaprant is effective both clinically and statistically significantly in the improvement in depression symptoms as measured with Hamilton D. But the striking effect, and this is given as a monotherapy, many of the agents that are tried for depression are tried as adjunctive. You keep an SSRI in place and you add it. Navacaprant allowed reversal, withdrawal from SSRIs, and then was then tested as a monotherapy, and it worked. Strikingly, there's a patient subset with anhedonia, the loss of enjoyment in something they formerly enjoyed. And this is a poor leading indicator for clinical response. And as a monotherapy, navacaprant produced, again, clinically and statistically significant improvements in anhedonia. And this was something that I regarded as a real upside. So where is navacaprant now? We're exploring at Scripps other areas of potential therapeutic applications. We have an ongoing collaboration with Eric Zarilla and Marissa Roberto looking at alcohol consumption, and they have been showing significant decreases in alcohol consumption in models of alcohol addiction. And so a variety of deep mechanistic studies are in progress at Scripps to flesh out whether this mechanism may have some uses in the treatment of alcoholism. I've mentioned the restoration of rapid eye movement sleep in neuropathic pain. This is an area that is of high interest to us in terms of understanding the mechanism. We have evidence that blocking the kappa opioid receptor blocks some of the issues associated with withdrawal, the dysphoria and the cravings that you see, for instance, with opiates, with methamphetamine withdrawal. So there's another reason to persevere. And obviously, post-traumatic stress disorder is another area where much more work needs to be done. So where are we now? Navacaprant has completed phase 2 for depression and anhedonia. It had an end of phase 2 meeting, and phase 3 has already started in 2024. We hope Neumora have announced that they will have the top line data on the first of the phase 3 studies announced in the second half of 2024. There are 2 other phase 3 studies in major depressive disease that will complete by mid-2025, allowing for a new drug application if the data support this in 2025. In addition, we showed the effects of Kappa agonists on causing thought disorders. Much more work needs to be done on the role of Kappa in thought disorders, and there is a phase 2 trial that will initiate in 2024 looking at bipolar disease, which is that mixture of depression and thought disorders. So all things being well, navacaprant has the potential to pay it forward. It can impact on patients potentially as early as 2026, and hopefully can then bring additional resources into the funding of fundamental research here at Scripps in that 2026 to 2040 timeframe. With that, thank you very much. Let me just acknowledge the members of the Rosen and Roberts labs shown here: Ed Roberts, Steve Brown, Nora Leaf, Miguel Guerrero, and our 2 newcomers, Suk Jung and Fiona. Fiona is being somewhat disembodied because she was on vacation in Colombia at the time we took the photograph. Again, we thank Frank Pareca, we thank our colleagues at Blackthorn Therapeutics, and we thank for the support the Blueprint Neurosciences, NINDS, the Molecular Libraries Initiative, that was the National Institute of Mental Health, and of course, Blackthorn Therapeutics. Thank you.
All right, so thanks to you. That was terrific. I always learn something, and I always appreciate learning a new word. And anhedonia is a word that was not. I was sort of perhaps more familiar with hedonia, but anhedonia is a new word. But I kind of wanted to think about that. So maybe I'll ask a real science question based on that. So what happens if you give a Kappa opioid receptor inhibitor to a normal person?
Interesting enough, that was phase 1, right? And the side effects were negligible. So there was no sedation, there was no alteration in mood, right? Because it doesn't deal with
There's no hyperhedonia.
There's no hyperhedonia. It removes a noxious stimulus, namely dynorphin, but it doesn't on its own necessarily inappropriately elevate mood. The only place we noticed a difference was in executive function if you stressed. So an experiment was done by the folks at Blackthorn where they looked at executive function. This is prefrontal cortex signaling to the amygdala, and Kappa is implicated. And if you agitate someone by giving them a benzodiazepine inverse agonist, in this case FG42, they become agitated and they lose the ability to focus on intellectual tests of working memory and executive function. If, however, you give them navacaprant, it blocks the ability of the GABA-A inverse agonist to induce that agitation that disrupts executive function. So I think it may also be useful, for instance, in a fighter pilot or a special forces operator who is subject to tremendously high pressures but needs to maintain executive function under the most stressful of circumstances. But I don't see any obvious downsides. In addition, we didn't see any sexual dysfunction.
So leading up to a question from somebody in the online audience, so can you use these compounds to intervene with drug-induced psychosis? Is there any application there?
The answer is we should test that. You know, in the absence of data, right, I'm just another guy with an opinion.
Yeah, right.
And that's, you know, these are the additional uses that are testable.
Very careful answer. Very careful. So I was trying to remember the salvia drug that you showed. That's also a kappa opioid receptor.
Agonist, yes. Agonist.
Oh, it's an activator.
Exactly.
Got it,
So you switch it on, you go bonkers. You switch it off,
right.
You're back to normal.
And so would your compound counteract that?
We know it does.
Okay.
We've done the binding assay.
Careful answer, but that's probably not going to be one of the applications. Now, so we have, let's take some questions from the audience. We have a lightsaber in the back. Please just.
Hi. Yeah, so my question is in regard to the developing process of the medication. In the developing stages, how do you predict and account for patients who, after using the medication for a long time, it just stops working?
You can't.
Okay.
You can't, right? You can only test for the periods of time that you test. So in this case, you're testing in phase 3 for 6 weeks, but you do a follow-on study. So patients that come off the 6-week study, whether they're on placebo or active, go into the follow-on study if that's what they choose, and will then be followed potentially for years. And that's how you build the safety and efficacy database as you do it. But no, the only way to know that is to have patients exposed for long periods of time and have patients come off and go back on again. And that's part of normal clinical development.
Anybody else? So I've got another online question. So you talked a little bit about some of the other potential applications, but what about, there's a few things like schizophrenia,
yep,
or obsessive compulsive disorder, and then patients that have a variety. You know, what if you have multiple disorders? So how do you parse this out, and are combination therapies possible?
Yeah, and the answer is that
I know the answer you're going to give, but I want to know what you think.
No, right now it's too early. Right now it's too early to pass it out. Mm-hmm. I mean, do we know that kappa signaling tone through dynorphin drives thought disorders? Yes. Do we know that is there evidence from patients that that's the case? Yes. Is there a peripheral surrogate marker that we could follow that might give us some insights? Quite possibly. So these things are knowable. And now that we've made tools, pharmacological tools that are safe and effective, we now have the ability to explore the wiring in more complex patient settings. But the initial studies always need to be very tightly focused, right, because you have to achieve safety and efficacy on defined indications, right, that matter enough to bring it to the market. Once it's on the market, many of these others become explored as a natural part of the evolution or life cycle of a compound.
So if you try to put too many indications in a clinical trial, then you're bound to fail on some of them. So you have to very carefully choose.
Exactly.
Okay, we're going to go after this. But now you're armed with, you'll have phase 3 data, so you know your compound is perfectly safe and effective. and you're saying you can just go after all of these other indications.
Correct, and then there are also investigator-initiated studies that explore these mechanisms further. So there are many opportunities, but the initial threshold is make something that's safe and effective that can actually answer the question, and that's the job of basic research.
So you're this is about 10 years behind ozanimod. And so how's that process of developing alternative indications gone for ozanimod?
What's actually been really gratifying with ozanimod in multiple sclerosis has been to see the long-term data, the 5-year data, and also the cognitive function data. So one of the things that ozanimod does by diminishing inflammation within the central nervous system is that it protects patients with multiple sclerosis from shrinkage of the brain. And we saw this initially on looking at the CT scans and the MRIs, and they were being protected from the thinning of the cortex and the loss of volume of key nuclei like the thalamus, for instance. And so as a result, they've gone on to do a study measuring cognitive function in patients on ozanimod or off ozanimod. And other mechanisms do not seem to provide the same protection. But ozanimod, at least at 2 years, is providing a protection, an enhancement of cognitive function. It suppresses a marker, neurofilament light chain, that is a sign of neurodegeneration and damage that's found in peripheral blood. And there's an improvement in cognitive function. So if you didn't make the compound that was safe and effective over the long term, this would never be known. But multiple sclerosis is not a silent disease, and patients should always strive to be on disease-modifying therapy to protect themselves from the long-term sequelae of the process.
So that's a good segue into, there's a question from the online audience about the Kappa opioid antagonist, and that is, so you can get through a clinical trial, but how do you know what the long-term effects are going to be for chronic medication?
Well, firstly, you have to have multi-year safety in animal species in order to be approved by the FDA. So that's, an important part of the process. And then you have the ongoing safety studies allowing long-term exposure for people that are monitored very, very closely that provide that. But I would also say that that I always like to see, a drug being exposed to tens of thousands of people, because once you've been in 10,000 people, in general, you have a better idea on how to use it.
So maybe, could you talk a little bit more about, you use the word or the phrase that we all use, it takes hundreds of millions of dollars to develop a drug. And then some part of that comes back. So we license these compounds to these companies and then royalties. So maybe just give us a sense for what was the impact of ozanimod royalties on developing the Kappa system, and then what are you going to do next?
So, clearly the ozanimod royalty has created a buffer that allows Ed and I to explore additional ideas, and it's a wonderful thing. We've also established the ozanimod fund that pays it forward to help support the strategic needs of our institution, which is also a very important part of building the fabric of community of scholarship, right, that makes Scripps so special. Our current focus is in is in the area of thought disorders. And we have a very active program now in collaboration with Kappa here at Scripps internally that is actually moving very, very nicely, knock on wood. And we hope that our next effort will actually be developed within the Kappa framework. And I can certainly say that as a faculty member, seeing how Kappa has augmented and facilitated our possibilities here within the institutional universe, it truly is a pretty amazing example of science changing life, not only for us as scientists, but also for patients on a broad scale. And I'm very grateful for that.
Yep, terrific. Well, thanks for that. And I hope the audience can appreciate that lightning has struck not once, but twice. And so we're pretty optimistic about our chances for a third strike. Thanks you. I think I just want to call your attention that we'll have a front row on May 15th, and it's going to be our colleague Jeff Kelly, who's another one of the Scripps faculty members who's actually discovered a medicine and brought it into treating patients. So please sign up and tune in for that. And as is my custom, you will find me on May 15th in the front row.